Hey guys, hope you're doing okay. This video is only for the online part of the lab. So if you're scheduled for a face-to-face meeting this week with me, you're not supposed to be seeing this video, but if you have an online lab scheduled for this week, you're in the right place. This topic is going to be the macromolecules one. It's a very interesting topic. Students always seem to enjoy this topic for some reason. It usually involves a lot of chemical testings and a lot of color changes. So it's a very visual lab and students seem to really enjoy it. So I hope you guys like it. Again, just to reiterate, if you're supposed to see me this week, you are not supposed to be seeing this video yet. Yeah, if not, there's my email. You can send me an email for any questions you guys have. Remember, I'm here for you. But yeah, let's go. I'll start with a very sad news. No quizzes for now, and not for the foreseeable future at least. I know. A lot of students, including some instructors, were having problems with the Top Hat portal, and we simply just went ahead and scrapped the quizzes. We have enough points in other activities, so we just went ahead and removed quizzes for now. I know it's very sad for you guys. It's very sad for me too. But yeah, just so you guys know, from now on, unless something changes, there are not going to be any more quizzes. Now, some helpful tips for today's investigation. Keep in mind, this video is a brief summary of what we will be doing today in the lab and some short explanations. If you want the long form explanation and the important data, it's found on the Canvas portal under the Macromolecules Online Lab tab. And then you will see the background information documents. And there you have more information about macromolecules and then some tables that you guys have to complete as part of the lab. But we'll get to that in the future. Now, what will we be doing during today's lab? What are the things that you need to do? First of all, we're going to identify and describe various types of macromolecules. Then we're going to determine what types of macromolecules are in what types of food. And finally, we're going to write an experiment summary. I'm going to refresh you guys' memory on how to write a summary. If you haven't read the document yet, I'm going to teach you guys then. And then comment with Flipgrid, which is something I'm going to explain at the end of the lab. Now, one of the most important macromolecules are carbohydrates, especially sugars. They can be either monosaccharides or disaccharides. Mono means one. So whenever you see the prefix mono, usually or almost always going to be one. So for example, monosaccharides, we have one saccharide or one sugar. And the prefix D, Di, means two. So for example, disaccharides means two sugars. They are used as a cellular food source. They are our fuel. So, you know, if you don't eat for a couple of hours or even a couple of days, you're going to feel groggy, you're going to feel sleepy, you're not going to have enough energy, simply because we are not consuming the fuel we need to give us energy. And if we wanted to see if something has sugars in it or not, we will then do a Benedict's reagents test. And if that thing turns orange or red, it means that it has sugars in it. So let's say if we got a piece of bread and we wanted to see if that bread had sugar in it or not, we'll just put some drops of Benedict's reagents. And if that bread turned red, it means it had it. And if it not, it means it doesn't have sugars in it. Another type of important carbohydrates are the starches. Starches are mainly found in plants, and they are a polymer, which means there are a lot of molecules joined together. So monosaccharides is one, disaccharides is two. Poly means a lot. So a lot of glucose molecules in this case joined together. They're used as structure and as an energy source in plants or an energy storage in plants. And their presence is indicated by iodine. And if it's a positive test, then that thing will turn black. So for example, potatoes. I know potatoes have starch in it. If we slice a potato in half and then we'll put a couple of drops of iodine in them, they'll turn black, letting us know that that potato has starch in it. A very important macromolecule are proteins. Proteins are in charge of almost everything in our body. Enzyme structure, transport, gene expression. A lot of important stuff are relegated to proteins. They can have highly folded structures, and depending on the type of structures that they have, they're going to have different functions. Again, this is something we're going to talk about very briefly here, but in class, you're going to hear more about proteins. And the presence is indicated by a Buret's reagents test. So if we wanted to see if something had proteins in it, we'll just put a couple drops of Buret's reagents. And if it turns purple, it has proteins in it. So if you had a protein shake, which I'm pretty sure has proteins in it, and then put some Buret reagents in it, the protein shake will turn purple. Cool. Lipids, fat, glycerol, fatty acids. They have hydrophilic heads and hydrophobic tails. So they're composed of two structures, the tail and the tail, sorry. The head is hydrophilic. Every word that ends in philic usually means lover. So hydrophilic means lover of water. So if the head is hydrophilic, it'll mean the head's gonna be always facing towards the water. It's gonna be looking for water. It's not gonna have any problems whatsoever with water being around. But hydrophobic, if something ends with phobic, it means scared or doesn't like. In this case, water, hydro. So if you have a hydrophobic tail, the tail's gonna be hiding. It doesn't wanna get wet. It doesn't like water. It doesn't mix with water. So for example, if you have oil, that you know what, that doesn't mix well with water, oil is hydrophobic. It's afraid of water. It won't mix with water. You have water, you put oil in it, the oil will stay on top, meaning it's hydrophobic. If it were hydrophilic, it will easily mix with the water, go inside, have a blast, whatever. It's used for energy storage and membranes. We're gonna have in our cell membranes a wall of phospholipids. So yeah, again, the process, you're probably gonna see it in class, not with me, but it's a very, very cool and interesting process. And if you guys have any doubts with it when you see it in class, come to me and let me know, especially for the cell membrane part. I really enjoy explaining that to students. And the presence of lipids or fats is indicated by Sudan's 4 reagent turning bright red. So if you want to see if milk or whatever has fat in it, drop a couple drops of Sudan 4 reagent, and if it turns bright red, it means that it has fat in it, or fatty acids, or glycerol. Sorry about that. Now for the next part, we're going to determine the macromolecules in common foods. So we're gonna get a couple of food items, and we're gonna do the different tests and see if it turns color, if it has it, or if it doesn't have that macromolecule in it. So on your lab background document, you're gonna be seeing a lot of tables depending on the food you're seeing. So when you see the video, like the experimental video, then Dr. Shah will be doing the test there, and you're gonna be seeing the different types of results, and you should be taking notes on those tables to see, oh, milk turned this, or hot dog turned red or black, whatever. So setting up an experiment, we are going to have a control group. Control group is super important. It will let us know if the reagents or the stuff we're working with are viable. So for example, let's say we have a potato. I know that potato has starch in it, and I have chicken, which I'm sure chicken doesn't have starch in it. And I wanted to see if the iodine I was working with was good or not, or maybe it's spoiled and it didn't work anymore. And a control group will let me know that. So for example, we got a positive control group, which contains only the molecule of interest. So we have a potato that I know has starch in it. I'm 100% sure it has starch in it. And then we have chicken, which I'm 100% sure doesn't have starch in it. And we're gonna test our iodine. If the potato turns black and the chicken also turns black, something's iffy there with the iodine, because I know the potato doesn't, I know the chicken doesn't have starch, so it's not supposed to be turning black. Or vice versa, if I put the iodine on the potato and it doesn't turn black, something's wrong again with the iodine we're using. So again, a control group is something that we do to make sure that the things we're working with are effective and are working properly. Positive control is the thing I know will react positively to it, and the negative control is the one I know that will not have some reaction whatsoever. Our experimental groups are the things we don't know. I mean, we don't know what types of macromolecules hot dog have, or tofu has, or energy drink has, or milk has. So those are the things we're going to be experimenting with on this lab. And in the experimental video you're going to be seeing, we're going to be going through hot dogs, tofu, energy drinks, and see what types of macromolecules they have using the test I just described to you guys. And again, the dependent and the independent variables, most of you guys, I would say 80 to 90% of you guys got it right on the B assignment. You were able to identify correctly the dependent and independent variables. But if you still have troubles with it, don't worry. I know it's hard. Happened to me too. Let me know. Just gonna explain it again very briefly here. But if you still have doubts, send me an email, or when we see each other in person, just let me know and I'll take my time to explain it to you. And this experiment, our dependent variable, that which we cannot control, are the types of macromolecules present. I cannot go to tofu and say, you will have proteins in it, and boom, out of nowhere, proteins, tofu is gonna have proteins, or say, tofu now has sugar. I cannot dictate to tofu what types of macromolecules they have in it. I cannot change that whatsoever. What I can change, what I can fiddle with, are the types of food that are going to be tested. I can choose hot dogs, I can choose chicken, I can choose tofu, I can choose a soda. That I can control. I can actually control which types of food I'm going to be testing. And then after I do those tests, I cannot control which types of macromolecules are going to be on those particular pieces of food. A thing and a reaction to that thing. Picking a hot dog and then seeing what reaction that hot dog has to that type of experiment. I cannot control their reaction. I can only see it. Please, if you still have doubts, let me know. Writing your hypothesis, again, a very, very brief summary of a hypothesis. Remember, it's a casual explanation. It has to be tested, but it doesn't necessarily have to be correct. So for example, a hypothesis, I can say a potato doesn't have starch. And it turns out that it did have starch. I was wrong. It's okay, it happens. You can have a wrong hypothesis. For example here, the Red Bull turns black in the presence of iodine because it contains starch. That could be true, but it could also be wrong. That's okay. For this lab, you're gonna write a hypothesis for each of the 4 foods being tested. So you will say milk has sugars in it and will turn bright red, or will turn red because it has sugars, or milk has proteins and it'll turn purple when I put the test in it. So again, just do the hypothesis. Don't worry if it's right or wrong. I will not be judging that. The hypothesis, it can be correct, it could be incorrect. Don't worry about it. It's a hypothesis. It's meant to be proven. So this is the lab procedure. For your lab summary, you're gonna be writing it as if you were in person. So if we were doing it in person, the first thing I'll tell you is step 1, label your tubes from 1 to 6. Then step 2, get your reagents, make sure they're properly labeled, put on your safety equipment, and then just transfer 2 milliliters of each test on each solution to the test tubes, and add the amount of reagents called for in the protocol. That's in your document. Then turn on the hot plate, boil some water so we can do the Benedict's reagents test. The Benedict's reagents test, the one for sugar, needs to be done in very, very hot water in order for it to be effective. And after we did all the tests, we're gonna clean our tubes and then use a soap brush and use just enough soap to fill the round end of the tube. And then we're gonna wash them with diluted water, and then we're just gonna put them out to dry. Again, this is a very basic structure of the procedure. In the lab summary, you guys are gonna go, okay, first we labeled the tubes, then we put there 2 milliliters of each reagent, then we did this, then we cleaned the tubes, and then we put them out to dry. That's the procedure. You can use this PowerPoint as a guide to write that procedure. And then after we did all the experimental things, we're gonna then write an experiment summary and a comment with Flipgrid. Displaying your data. When you guys finish collecting the data, remember the experiment will be done on a separate video, on a separate experimental video. You'll see Dr. Shaw doing the experiment, and you're gonna have your lab background and your lab report for that day, and you're gonna be filling it in as you see Dr. Shaw doing the video. After that, you're gonna end up with a lot of tables for each type of food. So what you're gonna do is you're gonna compile all that tables into one big final table, and that's what you're going to include in your lab summary. How is that final table gonna be structured? Well, we're gonna have our independent variable will be placed in the leftmost column, meaning that our independent variables, our types of food, are gonna be on the column hot dogs, milk, energy drink, tofu. And then our dependent variables are gonna be on the first or the top row. So we're gonna have sugars, starch, lipids, proteins, and then we're gonna fill the table off. So for example, milk, does it have proteins? Yes, no. Milk, does it have starch? Yes, no. Milk, does it have sugar? Yes, no. And then you're simply gonna do a very fun, nice table, and you're gonna put that in your summary. That's the only table I wanna see in your summary. I don't care about the other tables. The other tables, you're gonna use them to fill out the final table. Remember, the columns are gonna be the different types of food, and we're going to have on the top row the different types of macromolecules, and in the middle of the table, just yes or no if the macromolecule is present. If you guys wanna add the color, go ahead and do it. I'm pretty sure that on your main class, this is gonna come on the test. When I used to teach in Puerto Rico, this is something that we taught students, and on the test, I can tell you 100%, it always comes. It said, I had a piece of chicken and it turned purple. What does it mean? What type of molecule does it have? Or I have this type of thing, I don't know what it is, but when I put iodine in it, it turned black. What type of macromolecule does it have? Or vice versa. If I know something has proteins, what type of test should I do to see if it'll have proteins or not, and what is the positive color result for that type of macromolecule? So just please make sure you know the positive result color and what type of macromolecules trigger which type of test. But yeah, after that, you're done with the table, you're done with the experimental part. For the lab summary, go to the document I sent you guys via email last week, and it's still posted in Canvas. Is how to write a lab summary. It's a detailed guide. It's very easy to follow. You already even have the categories set for you, so you just put the different types of categories, procedures, conclusions, results. You have a filled out report on that, how to write your lab summary document. So you can use the same structure, and you can take the same type of writing style that they did and incorporate it into your document. Keep in mind that the lab summary is very, very short, usually 250 to 300 words. Shouldn't be more than 2 pages, including the table. So yeah, something that you can get it out of the way pretty easily. But if you have any doubts writing it, again, send me an email and I'll meet with you guys, or I'll send you an email back answering your questions. The lab summary should be uploaded to Canvas by noon on Friday, not midnight on Friday. If you guys make a late submission, the gradebook closes and I can't assign you points, even if for some reason you did it 1 minute late. The gradebook closes and I can't put points on your assignment. So please be sure to hand it in on time. If it's, you have the whole week, you have from Monday to Friday, so do it as quickly as possible, get it out of the way. If you have doubts, let me help you. If you think that you're having struggles with Excel, don't wait till the last minute, because probably Excel's gonna go all crazy on last minute and you are not gonna be able to submit it in. So yeah, please, please, please don't hand it in late, because I cannot give you any points for late submission. Canvas won't let me. And the last part of your summary should contain a Flipgrid post. We should address the following: your name, a brief summary, the most challenging part, something new you learned. Hey, I didn't know milk had sugars or whatever. And you're gonna be using the Flipgrid platform. So just put Flipgrid on Google. There's actually a Flipgrid tab on Canvas. Create an account. Use this join code right here, 78642DC4. And you'll automatically join my topic. You can use your cell phone. It's an app on your cell phone or in your computer. Just record yourself. Hey, my name is Fernando. In this experiment, we did this types of food. Again, you have it here on the PowerPoint. We tested this types of food for this types of macromolecules. The most challenging part was heating the water, whatever, getting the food together, or not being there in person. and something new you learn, like I said, hey, I didn't know milk had sugar or whatever. Yes, super brief, 1 minute or less. Don't worry about it, and you'll get free points for that. So again, it's free points very, very easily. So please, please do it. Yeah, other than that, we're done for today. If you guys have any other questions, important. When you do the Flipgrid post or when you do your Flipgrid sign in, be sure to sign in with the OU ID. All right, use the email OU gave you, like fernando.melendez1@ou.edu. Don't use a Gmail account. Don't use any other type of account. Use your OU account. Because that's the only type of account that's authorized to post in my Flipgrid. Yeah, if you have any problems with it, again, send me an email and I'll figure it out with you guys. Yeah, hopefully see you soon and stay safe. Go ahead and stop the recording. Again, take care. See you.